So I'll talk a little bit briefly today about computational tools for drug discovery and longevity, and a particular part which relates to actually simulating cells as well. So as I come from the computer software background, I got naturally interested in can we actually simulate biology. As I got into the space, I realized it's actually very complex. Like there's just so many genes, so many effects that it really can't be handled well without software. And ultimately, what we want to do is be able to understand biology and make changes and actually see what effects they have. And the reality is, as humans, we can only understand the principles. Everything else needs to be filled in with modeling and simulation. And for this, we really need both software and wet lab to collaborate. And ultimately, the results will be seen in the models and software. So we have this great progress in wet lab hardware tools. We got genomics revolution, multiomics is very active now, and we got bioinformatics and all the tools. And what now we are able to do is bring together the AI ML, but also physics and other tools into a more comprehensive model. And that's what I really love to see happen in a space in 5-10 years. So really, we need to systematically capture biology at all these different levels and integrate all of it. So we are working at Deep Origin and some of the infrastructure around doing that. But today, I'll talk more specifically about cells. And this has to do with simulating biochemistry and ultimately cells for drug discovery and longevity work. So really, we want to be doing is understanding how do molecules interact at more complex systems. And I'd love to be able to get to a point in space and time where we can take a young cell and an old cell and really contrast the two, see what the differences are, and actually do something about it. So I feel like we're at this space where actually approaching ability to simulate the living systems. We're still arguably far away, but lots of pieces are in place. And if we talk about the cell models, then you kind of have a range of technologies. You have physics, MD, and these tools have been existing for a while, but they are kind of too slow to actually get to the scale we need. And now AI is really filling in many parts. So really, if you want to have a model which can predict the phenotype and therefore you might be able to see if a disease changes and so on, then you want to take lots of high throughput experiment data, train models on it, but also build bottom-up models with physics and details and integrate these. And if we could do that, then we'd have this kind of technology stack where we can perhaps have molecular dynamics at the bottom, and coarse-grained models, AI data filling in, pathways, and then a full system. And if you actually have that full system, then you could take, let's say, a molecule, which is maybe a novel molecule, and actually introduce it and see how does it actually affect the whole cell. And you imagine, if you have a molecule, you need to know where does it bind, what is the kinetics, what pathways does it affect. Okay, and if this pathways are affected, how does the cellular structure change? So there's many different levels of answers. So I'll just talk a little bit about some of the technologies in the Deep Origin we're working on. This is really pieces which have existed in an industry, some things we're doing anew. And to give you kind of a cursory overview. So one thing is kind of classically people have tools for is ligand dogging and virtual screening. So you have your protein, you have your molecule. How does it dock? So docking has to do with basically fitting it within a protein shape. And surprisingly, technology has existed in some shapes or form for 20 years, but it's really not that good. And now with AI and high fidelity methods, we can actually get it to be a lot better. So in our case, the plot we are able to do on the order of 10x better than open source solutions. But by having this high fidelity docking, you can now take the compound and you can screen it on billions of compounds, and that usually requires AI because if you dock every single one times a billion, it's still expensive. And you can look at databases such as NME and e-molecules and others and actually be able to find the best binding. So, okay, now let's say we can bind a molecule to a protein. Well, what is the shape of a protein? How do these proteins interact? That kind of gets into the next level. And AlphaFold, you all know, made these drastic improvements and gave us shapes. But there's also challenges. Like, it's not really, as far as most people believe, it's not yet learning the physics of the matter, and so therefore it might not know how does the protein chain over time. Are there hidden pockets? If it's disordered, how do those tails interact? So there's other alternatives. One particular area we work on is building this coarse-grained protein folding model, which is physics assisted by AI. It has a fun name called AWESOME, Associative Memory Water-Mediated Structure and Energy Model. But basically what it does is protein structure prediction. It can do that, but also dynamics. And you can do dynamics of binding with DNA, lipids, and other things. And the basic premise here is instead of having every atom represented on a molecule, you have just a fewer beats. And of course, you need kind of a custom trained energy field, and then you can pull in bioinformatic data with AI, which help nudge it to actually get better results. So that's a possible coarse graining approach. Here's an example of such simulation running. And this is the optostatin venom protein binding to, it doesn't matter. But the point is, if you run a simulation, you can actually see the dynamics. So, and if you go a step higher, we get to the pathway models. And these, again, have existed for a while, but usually they're small. And this is, how does a gene affect a protein? And what is the whole sort of feedbacks to generate some outcome? So these type of models can predict how a cell responds to input. Will it grow or die? What are maybe effects of some mutations? So we'd love to get to a point where we can simulate every pathway and in a computer model. Now, right now, they're much smaller, but that's one of the areas, and it has benefits in oncology. And depending on what you look at, it can give you an extra tool compared to AI. So AI models might be able to do a lot of predictions, but there's cases where there's just not enough data, and having these physics-based models may give you another way to get more combinations tested. So kind of wrapping all this up, okay, if we had understanding of protein folding, if we combine this in perhaps different AI models, we have the pathways simulated. Well, perhaps then we can actually look at the broader structure. So we are talking about fibers, a cell. Like we have this prototype called Median, which is simulate actin myosin formation, and how does this affect the cell shape? So now if you could integrate this kinetics and other data into this coarse-grained model, then you can see how the cells grow and move and die, and it could applications for metastasis and other things. So that's kind of the vision, and which would require lots of companies. We're doing some of these parts, but I would love to see all these technologies come together in the next, let's say, 5 years would be pretty awesome. So that's it for me. There's a link to a newsletter. We write about some things we do at our company, and it has a little bit of humor in it. So,
thank you. I love how you went from 10 to 5 years in the end. Okay, we have time for a question. Question, comment.
Thanks, great talk. I was wondering if computation is a bigger bottleneck here or data is a bigger bottleneck to make these simulations more practical and, let's say...
It depends on the problem. I would say for AI, the data is a bigger bottleneck. For physics, it's computation. So molecular dynamics is very interesting, but the problem is anything which is like in millisecond scale, something of biological significance for a larger atom system is too slow.
All right, well, we have tons of time to get into it in the workshop, which are starting now. Thank you so much, Michael.